I'm Ron McGlennon, Medical Director of OralDNA Labs, and I'd like to welcome you to another installment of the OralDNA blog, Clinician's blog. Today, I thought we would take an exploration of a recently published article that brings to bear some new details about what we know about how periodontal disease relates to generalized systemic inflammation, which of course leads to a number of systemic disorders such as cardiovascular disease, some malignancies, diabetes, and so on. In an article published in the Journal of Dental Research from September 2020, the authors have described how polymorphonuclear leukocytes, or PMNs for short, are centrally involved in the signaling process leading to not only periodontal disease, but in patients who have other systemic inflammatory conditions, there is crosstalk and a synergism between that effect. The article is entitled Periodontal Inflammation Primes the Systemic Innate Immune Response. Let's first talk a little bit about the terms in the title. The immune system is described broadly in having two components, the innate immune response and the acquired immune response. For brevity, the innate immune response involves those things that happen very quickly in response to an inflammatory effect, a bacteria, a virus, a chemical insult. Where does that occur? It happens at the interfaces of our bodies and those parts of the environment, our mouth, our skin, our gut, and so on. Periodontal disease is an excellent model of this. Periodontal disease involves bacterial growth in the periodontal sulcus, and when those bacteria grow to a high enough level, typically due to poor oral hygiene, contributed to by our genetics, then we have the body responding with an inflammatory response. Cells that reside in the gingival epithelium are always surveilling what's going on in that periodontal sulcus. Those cells principally are macrophages, which release a series of cytokines that then recruit these innate immune response cells. In this study, the authors Fine, Chadwick, et al. Demonstrate that in a mouse model, you can induce periodontal disease, and in so doing, you mobilize the production of polymorphonuclear leukocytes, or PMNs for short, from the bone marrow to the site where that inflammation is occurring, namely the periodontal tissues. The enhancement of PMN production is noted in the bone marrow, but is not observed in the blood or in other tissues such as the peritoneal cavity. Now, in the same mouse model, we can also inject E. coli into the peritoneal cavity, thereby creating an artificial peritonitis. In animals that have peritonitis with commensurate periodontal disease, you see the promotion of PMN liberation from the bone marrow, blood, as well as at the site of the oral cavity and in the periodontal tissues. In this case, the response is synergistic, suggesting that there is an ongoing and significant contribution of the inflammation arising from the oral cavity. Now, this was also replicated in a human model. And what makes this paper so interesting is that in an artificial setting, taking patients who were volunteer to stop practicing good oral hygiene, and after about 3 weeks, they had early stages of gingivitis. In those individuals, they found higher levels of PMNs in the oral cavity, and from those measures that they could do by flow cytometry, evidence of it coming from the bone marrow. Blood levels, by contrast, did not elevate in these patients. Thereby, the human and the mouse models were similar. Now, what about these PMNs? How did they get the message to arrive in the periodontal cavity? Well, that remains a mystery, but studies also involved in this publication demonstrate that a panel of cytokines was investigated, and among those demonstrating to be elevated or enhanced were those including IL-6. IL-6 is known to promote granulocyte or PMN activation, and correspondingly, these PMNs also showed new antigen expression on the surface. In summary, this paper contributes significantly to the appreciation that periodontal disease is a source of chronic as well as episodic acute inflammatory effect. And that effect is played out by the promotion of polymorphonuclear leukocytes from the bone marrow, which then are signaled to arise and go to the periodontal tissues. One more piece of the puzzle of our understanding of systemic and oral disease connectivity. Thank you.